Good morning. I'm Jonathan. This is Conrad. This is Zinning. And today we'll be talking about human microsatellite DNA.
So the first topic we're going to review are the short tandem repeats, or STRs. These are microsatellites consisting of 1 to 6 repeating base pairs in the DNA. They're not known where they came from, but we suggest that they arose from slippage and mutations during replication. These have been used for human identification forensic cases. So this happens because every human has a different number of repeats in their alleles. So for example, on screen you'll see allele 5.2. This means that there are 5 repeats of the AATG nucleotides, and the 0.2 means that the second 2 nucleotides are repeated once again.
So polymerase chain reaction, or PCR, is a very powerful tool for amplifying DNA. There are a few key components. First of all, we need to have the DNA template, and this usually comes from the sample of interest. And then we have a forward and a reverse primer, which bind to the DNA template, and they allow the DNA polymerase to synthesize the rest of the strand. And the DNA polymerase uses nucleotides, so DATP, DTTP, DCTP, and DGTP to synthesize the DNA. The general procedure for a PCR is that the sample is first heated up to 95 degrees Celsius. This disrupts any of the hydrogen bonds between the double-stranded DNA. And then the sample is cooled to 50 or 60 degrees Celsius. This allows the hydrogen bonds to reform between the primers and the template. And then finally, it's heated back up to 72 degrees Celsius. This is the optimal working temperature for DNA polymerase. And typically, this whole procedure is repeated around 30 times. And each cycle doubles the amount of nucleotides. So after around 30 cycles, we'll have more than a million base pairs. And multiplexing is just a general term for the amplification of multiple loci at the same time. This saves a lot of time and money, so this is usually the preferred method of PCR now.
So after PCR, we can perform something called gel electrophoresis to compare and visualize the DNA migration across the gel. And so to do this, we'll need a casting tray with a negative electrode on one end and a positive electrode on the other end. Next, we'll need to add in a gel mixture of agarose and polyacrylamide, along with a comb suspended in the gel closer to the negative electrode. After the gel solidifies, we'll need to remove the comb and add a buffer solution and connect the 2 electrodes with the power source to generate an electric field that basically allows the DNA to migrate from the negative electrode towards the positive electrode. And this is due to the negative charge of the DNA molecule from the oxygen of the phosphate group. And after the whole setup, after we set everything up, we'll need to add the various STR samples into the wells of the gel. Next, we'll also need a loading dye, which impart color to the DNA so we can visualize it, and also sinks the DNA down so that the DNA can go across the gel.
So why are we teaching them all this?
Okay. So besides the fact that we're getting graded on this, last week someone stole all the lab practicals from Dr. Charvet's office.
Fortunately, the police arrived in time to collect a saliva sample from the criminal. Unfortunately, we were the 3 suspects because we were closest to the scene.
Okay, and so here are the results from the gel electrophoresis. So we color-coded the DNA fragments to match the color of the shirts of the suspects. And as you can see here, so clearly the DNA found in the crime scene matches the DNA fragment of the suspect with a green shirt. Wait a minute.
Who's you?
Stop! You're under arrest! Any last words? We finally caught you, you thief.
I would have gotten away with it too if it weren't for you meddling kids.
Thank you very much.